Okay, so now we have the second part of respiration where we're going to take our pyruvate and we're going to further break it down, release carbon dioxide, and eventually make some ATP, which is the whole point of doing this in the first place. So as you can see, I've already got my equation written on here: oxygen and glucose yielding carbon dioxide and water. The Krebs cycle is going to occur in the matrix, which is the innermost portion of the mitochondria, and it's all fluid. And this little box, that I drew on here is actually supposed to be the double membrane for the mitochondria. And there are some places where it kind of folds inwards, and those are called cristae, and that's where the electron transport chain occurs. So we're going to start with a preparatory reaction before the Krebs cycle actually begins. So we've got our pyruvate coming in. We're just going to follow one pyruvate at a time, although there's two pyruvate that get made for each molecule of glucose that gets broken down. So we have our 3-carbon molecule pyruvate come in, and we're going to take one of the carbons off along with an oxygen and release CO2 as waste. We're also going to lose a hydrogen to NADH. We're going to continue making electron carriers this entire time because those are going to be important in generating ATP. That leaves us with a 2-carbon molecule. Coenzyme A, which I'm just going to call CoA here, is going to attach, and it's going to usher that 2-carbon molecule into the Krebs cycle. So that 2-carbon molecule with CoA, and we actually call this entire molecule acetyl-CoA, it comes in, and CoA is going to help attach it to a 4-carbon molecule. That's going to give us a 6-carbon molecule. And the name of that actual molecule is citric acid, which is what makes fruit acidic. So we're going to break that down. We're going to take hydrogen from it to make NADH. And we're also going to release more carbon and oxygen as waste. That leaves us with the 5-carbon molecule. We're going to do it again. We're going to release some CO2 as waste. We're going to use some of the hydrogen from the molecule to make NADH. We're also going to make a little bit of ATP. That leaves us with a 4-carbon molecule, which we're going to rearrange by taking more hydrogen from it to make NADH, and also, make FADH2. And that 4-carbon molecule can then just join in for another round in the cycle. One thing I forgot to mention earlier, this CoA, after it'll usher that 2-carbon molecule in and help attach it to the 4-carbon molecule that's already existing. CoA will leave, and of course it's free to kind of go back around and pick up more 2-carbon molecules to usher in. So that's the end of the Krebs cycle. The whole idea was for us to make energy carriers. And they're going to move on. Our NADH and our FADH2 move on. We're going to use the electrons from that hydrogen and also the proton portion. You can split up each hydrogen atom into an electron and a proton. The electrons are used to power the electron transport chain. And the electron transport chain is going to use that energy to pump those hydrogen ions inside the cristae, in between the inner membrane and the outer membrane of the mitochondrion. So let me repeat that. We have an inner membrane and an outer membrane of the mitochondrion. Same thing here, inner, outer. We're going to fill this space between the two with hydrogen atoms, or hydrogen ions. And here's ATP synthase. We have all this potential energy here, and we're going to turn it into mechanical energy, and when it flows through ATP synthase, we're going to generate ATP. And it's a lot of ATP, 34. For each molecule of glucose, we're going to make 34 ATP. Now, all of these electrons that came from the electron transport chain have to go somewhere, and they are going to unite with some of these protons over here and oxygen to form water. Oxygen is known as the electron acceptor. It will accept all the electrons from the electron transport chain, combine them with the protons to form water as a waste product. And that's it. So we need to do a little bit of math for our summary. For the summary, let's look at the number of ATP that we made. So for glycolysis, remember that we had 2 net ATP. For the Krebs cycle and the preparatory step, we have 1. But remember that was just for 1 pyruvate, but there's 2 pyruvate for every glucose molecule, so it's actually 2. And for the electron transport chain, we have 34. So that gives us a total of 38 ATP for 1 molecule of glucose. Now for our FADH2, we made 1 for each pyruvate. There's 2 of them, so that gives you 2. And for our NADH, during glycolysis, we made 2. I'm just going to abbreviate. For glycolysis, we made 2, and for the Krebs cycle, we made, in the preparatory step, we made 1, 2, 3, 4, but again, that happens twice because we have 2 pyruvates for every glucose molecule, so that's actually 8. So that gives us 10 NADHs total. Each of those is going to give you 3 ATP each, and each FADH2 is going to give you 2, so that's 30 plus 4 gives you 34 ATP. So that's where that number comes from. And it's not always that exact number. Sometimes it can be less than that. But the whole idea behind this is to use the breakdown of pyruvate, which was at one point glucose. We're going to use the breakdown of it to make our energy carriers, which then go to the electron transport chain and generate all of our ATP, or almost all of the ATP, for the cell. And that's about it.